Welcome to my channel. Today, we're diving into the fascinating world of biology with 10 mind-blowing facts that will leave you in awe. From gene editing technology that sounds like it's straight out of science fiction to creatures that can survive in the vacuum of space, get ready to explore the incredible diversity and complexity of life on Earth. Let's get started. First, now imagine being able to go into a cell and precisely snip out a tiny piece of DNA, like fixing a typo in a book. That's essentially what CRISPR-Cas9 allows scientists to do. This revolutionary gene editing technology is transforming the field of biology, offering the potential to cure genetic diseases, enhance crop yields, and develop new disease-fighting strategies. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which might sound like a mouthful, but it describes a special kind of DNA sequence found in bacteria. These sequences are like a bacterial immune system, storing snippets of viral DNA so they can recognize and fight off future infections. Scientists figured out how to harness this bacterial system to target and cut specific DNA sequences in any organism, including humans. It's like having a pair of molecular scissors that can be programmed to make precise changes in the genetic code of life. The potential applications of CRISPR-Cas9 are truly mind-boggling. Scientists are exploring its use in treating genetic disorders like cystic fibrosis and sickle cell anemia. It could also lead to more resilient and nutritious crops, reducing our reliance on pesticides and improving food security. And the ability to edit viruses could revolutionize how we combat infectious diseases like HIV and even cancer. Second, imagine being able to get all your energy from the sun, just like plants do. Now you're probably thinking, that's impossible. We're not plants. Well, prepare to be amazed by the Elysia chlorotica, a sea slug that's breaking all the rules. This incredible creature has figured out how to steal the secrets of photosynthesis, essentially turning itself into a solar-powered animal. Elysia chlorotica, also known as the Eastern Emerald Elysia, is a bright green sea slug found along the Atlantic coast of North America. It feeds on a type of algae called Bacterial littorea. But instead of completely digesting the algae, it retains the chloroplasts, the tiny green organelles responsible for photosynthesis. The sea slug incorporates these stolen chloroplasts into its own cells, specifically into the cells lining its digestive tract. This process, called kleptoplasty, allows the sea slug to directly utilize sunlight to produce energy, just like a plant. Now, you might be wondering, how long can this sea slug survive on sunlight alone? Studies have shown that Elysia chlorotica can live for months without eating as long as it has access to sunlight. That's like you or me being able to survive on nothing but sunshine for months. This remarkable adaptation highlights the incredible ways that life on Earth can evolve to thrive in diverse and challenging environments. Third, get ready to meet the microscopic superheroes of the animal kingdom, tardigrades. These tiny creatures, also known as water bears or moss piglets, are renowned for their incredible resilience and ability to survive conditions that would be lethal to most other life forms. Tardigrades are typically less than a millimeter long and can be found in a variety of habitats around the world, from the depths of the ocean to the tops of mountains. They're most commonly found in mosses, lichens, and soil, where they feed on plant cells, algae, and even smaller invertebrates. But what makes tardigrades truly extraordinary is their ability to withstand extreme environments. They can survive being frozen solid for decades, heated to temperatures above the boiling point of water, exposed to intense radiation levels that would kill a human, and even the vacuum of space. So, how do they do it? Tardigrades have a remarkable ability to enter a state of suspended animation called cryptobiosis. In this state, their metabolic processes come to a near standstill. They can dehydrate, retracting their head and legs and forming a tiny barrel-shaped ton. Their metabolism drops to less than 0.01% of normal, and they can remain in this dormant state for years, even decades, until conditions improve. Fourth, we all learned in biology class that genes are passed down vertically, from parents to offspring. But in the microbial world, things get a little wilder. Bacteria have this incredible ability to exchange genetic material horizontally, meaning between individuals that are not directly related. It's like you shaking hands with someone and suddenly acquiring their ability to play the piano. This process, known as horizontal gene transfer, or HGT, plays a significant role in bacterial evolution and adaptation. It allows bacteria to acquire new traits much faster than they could through traditional mutation and selection processes. There are three main mechanisms of HGT transformation, transduction, and conjugation. In transformation, bacteria can take up free DNA from their environment, often released by dead bacterial cells. Transduction involves the transfer of genetic material through viruses that infect bacteria. And in conjugation, bacteria can directly transfer genes to each other through a pilus, a hair-like appendage that acts like a bridge. HGT has profound implications, especially in the context of antibiotic resistance. Bacteria can acquire genes that make them resistant to antibiotics, and through HGT they can rapidly spread this resistance to other bacteria, even those of different species. Fifth, imagine losing a limb and having it perfectly regenerate, just like a starfish regrowing an arm. While this level of regeneration might seem like science fiction to us, it's a reality for some incredible creatures in the animal kingdom. These animals possess extraordinary regenerative abilities that have captivated scientists for centuries. One such creature is the axolotl, a type of salamander native to Mexico. These aquatic creatures are renowned for their remarkable ability to regenerate lost limbs, tails, and even parts of their heart, spinal cord, and brain. If an axolotl loses a limb, it doesn't form a scar. Instead, it regrows the limb perfectly, complete with bones, muscles, nerves, and skin. Another champion of regeneration is the planarian flatworm. These tiny creatures, typically found in freshwater habitats, possess an almost unbelievable ability to regenerate. You can cut a planarian into multiple pieces, and each piece will develop into a complete, fully functional individual. Scientists are diligently studying these regenerative marvels, hoping to unlock the secrets behind their extraordinary abilities. Understanding the molecular mechanisms that govern regeneration in these animals could have profound implications for human medicine. Sixth, get ready to be dazzled by the magical world of living light. Nature is full of surprises, and among the most awe-inspiring is the ability of certain organisms to produce their own light or manipulate light from their surroundings. These phenomena, known as bioluminescence and biofluorescence respectively, create some of the most stunning visual displays in the natural world. Bioluminescence is the emission of light by a living organism through a chemical reaction. This process typically involves a light-emitting molecule called luciferin and an enzyme called luciferase. When luciferin reacts with oxygen, it releases energy in the form of light. One of the most iconic examples of bioluminescence is found in fireflies. These nocturnal beetles use their flashing light patterns for communication, primarily to attract mates. The synchronized flashing of fireflies is a mesmerizing sight, especially on warm summer nights. Biofluorescence, on the other hand, is the absorption of light at one wavelength and its re-emission at a longer wavelength. Unlike bioluminescence, which involves a chemical reaction, biofluorescence is a physical phenomenon. Certain organisms possess specialized molecules that can absorb light energy and then release it as a different color. Seventh, bacteria might be tiny single-celled organisms, but don't let their size fool you. They're incredibly social creatures and have developed sophisticated ways to communicate with each other. This bacterial chatter, known as quorum sensing, allows them to coordinate their behavior and act as a collective, much like a multicellular organism. Quorum sensing relies on the production and detection of signaling molecules called autoinducers. Bacteria release these autoinducers into their environment, and as the bacterial population density increases, so does the concentration of these signaling molecules. When the concentration of autoinducers reaches a critical threshold, it triggers a response in the bacterial population. Bacteria possess specific receptors that can detect these signaling molecules, and once the threshold is reached, it activates a cascade of cellular processes, leading to coordinated changes in gene expression. Quorum sensing allows bacteria to engage in a variety of group behaviors, including biofilm formation, virulence factor production, and bioluminescence. Biofilms are complex, structured communities of bacteria that can adhere to surfaces, making them more resistant to antibiotics and environmental stresses. Virulence factors are molecules produced by bacteria that contribute to their ability to cause disease. Eighth, for a long time we thought our genes were our destiny, that our DNA sequence dictated everything about us. But in recent decades, scientists have uncovered a hidden layer of complexity within our cells, epigenetics. This fascinating field is revolutionizing our understanding of how genes are regulated and how environmental factors can influence our health and even the traits we pass down to our children. Epigenetics literally means above genetics, and it refers to changes in gene expression that don't involve alterations to the underlying DNA sequence. Think of your DNA as the hardware of a computer and epigenetics as the software that controls how that hardware is used. Epigenetic changes can occur throughout our lives and are influenced by a variety of factors, including diet, stress, exposure to toxins, and even social interactions. These environmental cues can trigger chemical modifications to our DNA or to the proteins that interact with DNA, ultimately influencing which genes are turned on or off. One of the most well-studied epigenetic mechanisms is DNA methylation. This process involves the attachment of a methyl group to a DNA molecule, which can effectively silence a gene, preventing it from being expressed. Another important mechanism is histone modification. Histones are proteins around which DNA is wrapped, and chemical modifications to these histones can alter how tightly DNA is packaged, influencing gene accessibility. Ninth, imagine a world where scientists can design and build entirely new life forms from the ground up, like engineers constructing complex machines from individual parts. This might sound like science fiction, but it's the ambitious goal of synthetic biology, a rapidly emerging field that's blurring the lines between biology and engineering. Synthetic biology aims to apply engineering principles to biological systems, designing and constructing new biological parts, devices, and systems. It's like taking the building blocks of life, DNA, proteins, and cells, and using them to create novel biological entities with desired functions. One of the key tools in the synthetic biologist's toolbox is DNA synthesis. Scientists can now synthesize DNA sequences in the lab, allowing them to create custom-designed genes and even entire genomes. This ability opens up incredible possibilities for creating organisms with enhanced or entirely new capabilities. The potential applications of synthetic biology are vast and span a wide range of fields, from medicine and agriculture to energy and environmental remediation. Scientists are exploring the use of synthetic biology to develop new biofuels, produce pharmaceuticals more efficiently, create biosensors for environmental monitoring, and even engineer bacteria that can break down pollutants and clean up toxic waste. 10th, get ready to journey into the fascinating world of the microbiome, the vast and diverse community of microorganisms that live in and on our bodies. These tiny organisms, primarily bacteria but also fungi, viruses, and other microbes, outnumber our own human cells and play a crucial role in our health and well-being. Think of your body as a complex ecosystem, home to trillions of microscopic inhabitants. These microbes, collectively known as the microbiome, reside on our skin, in our mouths, and throughout our digestive system, with the highest concentration found in our gut. The gut microbiome, in particular, plays a vital role in our health, influencing everything from digestion and nutrient absorption to immune function and even mental health. The bacteria in our gut help us break down complex carbohydrates that we can't digest on our own, produce essential vitamins, and compete with harmful pathogens, preventing them from taking hold. Research on the microbiome is exploding, and scientists are continually uncovering new and surprising ways that these microbial communities influence our health. We now know that imbalances in the gut microbiome, known as dysbiosis, have been linked to a wide range of health conditions, including obesity, type 2 diabetes, inflammatory bowel disease, allergies, asthma, and even neurological disorders. Thanks for watching. If you enjoyed this video, don't forget to like, share, and subscribe for more amazing content. From the gene editing revolution of Crispr-Cas9 to the mind-boggling resilience of tardigrades and the hidden world of the microbiome, biology continues to amaze and inspire. See you next time!